Dear learners, welcome back to NPTEL online certification course on novel drug delivery systems. I am Dr. Satish Devanapalli, working as assistant professor at Department of Pharmaceutical Sciences and Technology, Institute of Chemical Technology, Mumbai. So, in our previous lecture, we have discussed on various types of vesicular drug delivery systems. In that, we have learned what are the vesicular drug delivery systems. We have seen what is the importance of vesicular drug delivery system in a formulation sector. In that vesicular drug delivery system, we have seen what are the neosomes, and also we have learned the importance of neosomal formulations, and then we have seen methods of preparation, and also we have seen what is the difference between liposomes and neosomes. With that, we have already discussed on the what are the ethosomes, and ethosomes we have seen the differences between liposomes and the ethosomes, and how ethosomes can be utilized in a topical formulations, especially as in a transdermal drug delivery and cosmetic formulations. In the ethosomes, we have already discussed on the various mechanisms which help in carrier systems. In ethosomes, we have studied the mechanism. One is ethanol effect, and also we have seen ethosomal effect. So, how this effect will help in the penetration of the active molecules. So, today we are going to see on the remaining carrier systems. So, here we are going to discuss a brief what are these vesicles and the importance and the mechanism and the components which are involved for the preparation of the vesicular systems. So, in that we will start with the transferosomes, then we will see the bilosomes and the virosomes, and then spingosomes, and last one is the phytosomes. These phytosomes we will discuss with the one case study. So, here we will see the structure of the transferosomes, which are similar to the liposomes, but there are the other components which will help in the penetration. So, transferosomes are composed of phospholipids and edge activators, which are membrane softening agents. Now, by seeing the structure, it looks similar to the liposomes, but here you can see there are the edge activators which are represented in yellow color. So, that will help an additional function when compared with the liposomes. Transferosomes, we can split, we can divide this word into two words. One is a transfer, nothing but to carry something, zomes are nothing but body. So, these are the bodies, these are the structures, these are carrier systems which will carry the molecules from one place to another site, which is called as transferosomes. So, now here one of the important component is nothing but edge activators, which are example is twin 80 and the span 80, and in some other cases we use also. sodium cholate, which will help in the softening of the outer membrane of the vesicle. So, how this will help and where we can use such type of the transferosomes. So, now we will see how it will act within a pictorial representation. We will try to understand how the transferosomes works on the body. So, edge activators act as a membrane destabilizing factors which will destabilize the membrane to increase the deformability of vesicle membranes. When combined with the appropriate ratio with the lipids and edge activators, that optimum mixture, optimum ratio will convert this transferosome into the ultra flexible vesicles. That means self squeezing mechanism will help for the permeation. So, that is called as deformation, which results in the higher permeation capability. So, which are called as edge activators. They have the specialized function in the transferring of the molecules through the various cellular membranes. Now, with this picture representation, we will see how this edge activator will help in the penetrating from the skin layers. Now, here you can see, so this is the edge activator containing vesicles which are transferosomes. After administration, because of these edge activators, as we know, which will soften the membranes, which will destabilize the membranes. So, it will undergo the squeezing when between the cellular gaps. So, you can see the self squeezing mechanism. So, they will come into the thin structure and which is a deformable, then with that gap between these cells, so which will pass from that the cell structures. So, after that, once it is crossed through this cell layer, again it form into its original structure because of only with the edge activators. The reformation, you can see here is the deformation and the reformation after penetration of the stratum corneum. When transferosomes reach the skin pores, they can change their membrane flexibility and passing through the skin pores spontaneously. This is the so called as self optimizing deformability, and this property only purely comes with the edge activators. If you would like to read more on these transferosomes, I recommend to read this review paper, which is a open access review paper. Now, another vesicular systems, that is Bilosomes. Now, all these vesicular systems, the first term speaks about the component, right, like ethosomes, which contains ethanol. In other case, neosomes, non-ionic surfactants. So, now here when it comes to the Bilosomes, the name itself speaks about it contains bile salt stabilized nanovesicles, which are also known as Bilosomes, which are closed vesicles similar to the liposomes, which contains lipids, non-ionic surfactants and also the bile salts. See, one more additional component is coming. So, whenever new ingredient or new component comes, they must have some special application in the formulation. So, Bilosomes provide a significant protection from numerous faced burdens in especially oral application because of this acidic pH in the GIT and other enzymes presented in the GIT environment which will degrade your active molecules. So, that is why these bilosomes are preferable in terms of oral application when you want to take any antigens or when you want to take any protein molecules which are sensitive to the acid and as well as enzymatical conditions. Such cases you can take in a bilosomes, you need to encapsulate into these bilosome vesicles which will protect from the all type of the degradation. And the great thing that you can take hydrophilic drugs, even hydrophilic drugs similar to the other vesicular systems. And not only that, these bilosomes you can also use it for the transdermal drug delivery because of their fluidizing effect similar to the transferosomes and other vesicular systems. Now, you can see the structure which is similar to the liposomes, but here you will see some are the bile salts which will prevent the all type of degradation that will improve the stability of your formulations. So, now these are the various examples which are the bile salts, we can use it to make the bilosomes. Here we have one more vesicular systems which are virosomes. It is also known as virus like particles, also viral nanoparticles which are similar to the liposomes, but this liposome structure which is conjugated with the various virus based glycoproteins. So, that is why the structure when you see the viral particle, it looks like a virus, because we will extract the glycoproteins from the virus and then we will conjugate with the lipids and then we will utilize for the applications. So, here the major components are the glycoproteins which are viral components. So, that is why it is called as a virosomes. It looks similar to the virus. So, what is the use of the virosomes? To deliver any type of antigens to the host cell. So, what we do? We need to take the glycoproteins and need to conjugate with your lipid, so that it will also mimic similar to the virus. Now, you can see one example here. Here is the influenza virus which contain hemagglutinin as a one glycoprotein, and it contains another enzyme that is called as neuraminidase. This both will help in the conjugating, or you can say the binding on the surface of the cell, and neuraminidase will help in the cleaving after replication, cleaving from the host cell. So, when you take such molecules, you can see here. So, this is the virus. Now, if the virus need to the bind on the surface of the host cell, this is a host cell. So, this virus need to bind on some of the chemical constituents which are sialic acid. To bind on that sialic acid, so the virus should contain hemagglutinin, so that which can easily interlink with the cell surface. So, now neuraminidase will help in the binding of that cell surface, and hemagglutinin will help in the cleaving of the virus from the surface of the host cell. So, with this we will understand both hemagglutinin, neuraminidase will help in the replication as well as the release from the host cell. So, with this in the structure you will understand, see the virus required hemagglutinin and the neuraminidase. So, here you can see the hemagglutinin will helps in the binding and the internalization on the surface of the host cell. So, here the neuraminidase, this enzyme will help in the hydrolysis. So, once hydrolysis done, this viral particle, the new viral particle will release from the surface of the host cell. So, same mechanism we will use in the development of the virosomes. So, now you can see here how we will prepare a virosomal preparations. So, here we need to first get the our glycoprotein from the virus structure, okay. But here you can see the various microbial organisms. So, here you can see the virus and these are desired proteins, neuroplastic or tumor proteins and the bacteria and the parasite. So, you need to extract the desired antigen by killing the native microorganism. So, in this case we need to kill the virus or we need to remove the nucleocapsid, which is the major thing for the infection. Once you remove the nucleocapsid, you need to collect the viral envelope proteins and then that need to be reconstituted with your lipid. So, you will get in a liposomes and after that you need to encapsulate with your antigen. So, that will help for the enter into the host cell. So, with this you will understand first step is selection of desired surface antigen. So, with the desired microbial species you need to extract the antigen of interest. After that collect that antigen and select your required lipid and make a conjugation with this your antigen and then lipid. And after that by using any simplest general preparation methods you can make liposomes. So, which will give the virosome with antigens. So, you can complex or you can put in covalent bonding with your liposomes that will help to enter into the host cell similar to the virus. So, here one more vesicular systems which are spingosomes. Now, the name itself speaks that which contain the spingolipids. Now, the conventional lipids are replaced by the spingolipids. Now, here we will see what is the definition of that spingosomes. Spingosomes are concentric bilayer vesicles where an aqueous volume is entirely enclosed by a membranous lipid bilayer mainly consisted of natural and synthetic spingolipids. So, natural lipids are replaced by the spingolipids, so which are called as spingosomes. So, the major component of the spingosomes is nothing but spingolipids which are replaced from the liposomal structures. So, yes, they have a very new functionalities in their formulations. They have various advantages compared with the other vesicular systems. So, spingosomes are very stable to acid hydrolysis, have better drug retention characteristics. So, whenever you want to load any acid sensitive molecules for even oral application, such cases you can use spingosomes. Now, here are the spingolipids which are prepared similar to the liposomes, which are made up of spingolipids and the cholesterol similar to the liposomes. Yes, for the oral application are the one of the major application in the spingosomes. Now, these are the various spingolipids, these are the different sources. From that we can get these are all are natural sphingolipids, which we can utilize it for the application of the sphingolipid formulations. Now, we have in our phytosomes, which are also known as herbosomes. Now, we know phyto are nothing but natural, or you can say the plant based phytoconstituents. So, which will also show the activity in the human beings. So, these are the phytosomes are one of the growing nanotechnologies that can be used to improve the miscibility of bioactive phytoconstituents in lipid rich barriers, overcome their poor bioavailability. Now, we know that phytoconstituents comes in two categories. One is in the as in a water soluble, and other thing is water insoluble format. So, both are difficult in terms of the bioavailability as in a dosage form. To improve the solubility and also to improve the permeability, we need to play various formulation strategies to improve the solubility as well as permeability. So, that ultimately we will see the enhancement in the absorption that leads to the higher bioavailability. So, such phytoconstituents which are water soluble and water insoluble, such constituents we can put in a vesicular systems which are called as phytosomes. And yes, these are all our herbal source active constituents which are also known as herbal based bodies which are called as herbosomes. So, with this we can enhance the bioavailability of phytoconstituents. Now, here one more example, water soluble phytoconstituents like flavonoids, tannins, glycosidic glycans which are water soluble, but their permeability is very less, which cannot pass through the cellular membranes because of their large molecular size, which cannot be absorbed by passive diffusion due to their lipid solubility. Now, to improve the lipid solubility or to improve the permeability, you have to must load your water soluble active constituent in the lipids, so that the outer lipid will help for the penetration through the cell membranes. So, these are the several limiting factors in the crossing the cell membrane, similar to the pharmaceutical active ingredients. So, these lipids will help us to cross the membranes, so that can overcome the poor bioavailability. Now, you can see here, this is a picture representation. With the medicinal plant, you can extract your active constituents, that is phytoconstituents, which may be water soluble or water insoluble. To get the formulation, you need to convert this into the carriers, which are phospholipid carriers. Now, after that, you can convert that into the various dosage forms, either in liquids, lipids, in the oral solid dosage form, you can also convert into injectors, injectables, syrups and the powders. And that active constituent you can give it as a supplement for the treatment of various infectious diseases, and also you can put these molecules as an active and the passive targeting in the for the treatment of cancer cells. Especially the curcumin is a water insoluble molecule. So, by using the phytosomes, you can enhance the solubility, and that can be utilized. It is a well known, and the existing literature suggests that this curcumin is anti-cancer active molecule. So, such cases you can target to the cancer cells by either active targeting or passive targeting, which can also treat the cancer cells. So, here we are going to see one case study for all vesicular systems. So, with that one system I have selected to understand the importance of the phytosomal or vesicular systems. So, here is the reference. So, here the authors have selected the silymarin, which is a phytoconstituent which derived from the this species, which is well known, documented that silymarin is the hepatoprotective agent, but which suffers with the low bioavailability due to water solubility limitation. So, these authors, they prepared phytosomes with the various phospholipids by using the solvent evaporation techniques. They have optimized the formulations, they have done the in vitro studies, in vitro release studies, and some other encapsulation studies. Also they have seen the particle size, and after that they tried to see the efficacy of the plain drug, the pure drug of silymarin, with the optimized formulation. So, for that, before going that, so they have seen the even the bioavailability of the molecule in the rat model. So, now you can see in the figure. So, the blue color represents with the plain drug silymarin. So, this side is the plasma concentration and versus time. So, the plasma concentration with the pure drug, which has a very less amount of the drug we can see in the picture representation, whereas which is converted into the phytosomes, silymarin phytosomal formulation, which showed the maximum plasma concentration after oral administration of the phytosome based formulation. Now, after that they have seen the hepatoprotective activity in the rat model. So, before inducing the hepatotoxicity, so they treated this rat with the pure silymarin and then silymarin loaded phytosomes with the 100 mg per kg per oral dose. So, interestingly you will see the silymarin, pure silymarin dosage form, which is not showing major hepatoprotective action compared with the formulation based silymarin. So, after administration, after pre-treatment, you can read here the silymarin phytosome pre-treated rats shown the restoration of the cytoplasm and the central vein compared with the plain drug. So, here you will see the degradation, fatty degradation and the tissue damage after inducing with the carbon tetrachloride, which is induces the toxicity on the liver cells. So, whereas pre-treated with the phytosomal silymarin, which is protecting the from the toxicity of the carbon tetrachloride. So, with this phytosomal formulations are very efficient compared with the plain drugs. So, any type of active molecules can be converted into the phytosomal formulations, which will definitely show the higher bioavailability as well as the improved action we can expect from these formulations. So, with this I will conclude today's session. So far we have discussed on various vesicular systems from the neosome, transferosomes, ethosomes, bilosomes and virosomes, and also we have seen spingosomes, and at the end we have seen the phytosomes. In our upcoming classes we are going to see various other types of the nanocarrier systems. Thank you so much.